Induced mutation. The artificial production of mutation is called mutagenesis and the mutation inducing agents are called mutagens. Mutagens act at the molecular level, inflicting abnormal alterations on DNA. Mutagens are of two basic kinds, namely physical mutagens and chemical mutagens. In order to increase the frequency of mutation in experimental organisms, researchers often treat them with high doses of chemical mutagens or expose them to ionizing radiation. Mutations arising in response to such treatments are referred to as induced mutations. Generally, chemical mutagens induce point mutations, whereas ionizing radiation give rise to large chromosomal abnormalities. Physical mutagenesis. Physical mutagens mainly include the different kinds of radiations of the electromagnetic spectrum and also the products of the decay of the radioactive isotopes such as carbon 14, tritium or H3, S35, etc. The former includes short wave and high energy radiations such as UV rays, X-rays, gamma rays and cosmic rays. The latter includes alpha particles, beta particles and high energy protons and neutrons. Physical mutagens are two kinds, non-ionizing and ionizing. Non-ionizing radiation. Non-ionizing radiations include the UV rays which range from 180 to 400 nanometer of the electromagnetic spectrum. UV rays generate a number of photoproducts in DNA. Two different lesions that occur at adjacent pyrimidine residues, the cyclobutane pyrimidine photodimer and the 6,4 photoproduct have been most strongly correlated with mutagenesis. These lesions interfere with the normal base pairing, hence induction of the SOS system is required for mutagenesis. The insertion of the incorrect bases across from UV photoproducts is at the 3' position of the dimer and more frequently for 5' CC 3' and 5' TC 3' dimers. The cytosine to thymine transition is the most frequent mutation, but the other base substitutions called transversions and frame shifts also are stimulated by UV light, as are duplications and deletions. This figure describes the structure of a cyclobutane pyrimidine dimer. It also describes the structure of the 6,4 photoproduct. The structure forms most prevalently with the 5' CC 3' and 5' TC 3' between the C6 and the C4 positions of the two adjacent pyrimidines. The reversal of the mutagenic effect of UV radiation by visible light is called photoreactivation. The absence of inadequacy of the DNA repair mechanism in human beings leads to an autosomal recessive skin disease called xeroderma pigmentosum. They develop skin cancer in those areas of the body that are uncovered and directly exposed to UV radiations. Ionizing radiations. Ionizing radiations include alpha, beta and gamma radiations, X-rays, protons, fast neutrons and slow neutrons. Of these, fast neutrons, thermal neutrons and X-rays are commonly applied for mutagenesis in plants. All of them are highly penetrating. They consist of highly energetic photons which can penetrate the cells and knock down the electrons of cellular atoms out of their orbit, leaving charged ions or free radicals. This results in the ionization of atoms. Here in this table, you can see some of the mutagenic ionizing radiations. Gamma rays, they are basically photons in nature and the source is radioactive elements. X-rays are also photons in nature and the source is X-ray machines. Beta rays exist as free electrons and the source is radioactive elements. Protons exist as hydrogen nuclei and the source is cyclotron. Alpha rays, the nature of alpha rays is helium nuclei and the source is radioactive elements cyclotrons. Neutrons, Neutrons exist as neutral particles and the source is cyclotron. Ionizing radiation results in the formation of ionized and excited molecules that can cause damage to cellular components and to DNA. Many different types of reactive oxygen species are produced, including superoxide radicals such as OH. The most biologically relevant reaction products are hydroxyl radical OH, superoxide radical O2- and hydrogen peroxide. These species can damage bases and cause different adducts and degradation products. Ionizing radiation can cause breakage of the N-glycosidic bond, leading to the formation of apyrimidinic or apurinic site, and can cause strand breaks that are responsible for most of the lethal effects of such radiation. Ionizing radiation can cause both gene mutations and chromosome breaks. Chromosome breaks may result in structural aberrations such as deletions, duplications, inversions and translocations. In general, the rate of gene mutation is directly proportional to the radiation dose. This linear relation between radiation dose and mutagenesis indicates that a single radiation hit is enough to produce a gene mutation. The efficiency of effectiveness of radiations in mutagenesis is related to several factors such as stage of cell division, cellular level of oxygen, temperature, moisture content of the cell, etc. Cells in the diplonema and dikinesis stage of meiosis I are more prone to radiation induced mutations. Higher concentration of oxygen and reduced water content are also ideal for mutagenesis by irradiation. Chemical mutagenesis. A good number of chemicals are reported to be mutagenic. Many agricultural chemicals such as insecticides, fungicides and herbicides are found to induce gene mutations as well as chromosomal aberrations. The important chemical mutagens include base analogues, alkylating agents, acridine dyes, deaminating agents, hydroxylating agents, etc. Of these, base analogues act only on replicating DNA and the others act on both replicating and non-replicating DNAs. Base analogues. Base analogues are compounds very similar to nitrogenous bases. They are the derivatives of normal bases, so they can be used to replace the normal bases in the DNA for producing transitional substitution mutations. Artificially synthesized base analogues are 5-bromouracil, 5-chlorouracil, 5-iodouracil, 2-aminopurine, 2,6-diaminopurine, etc. Of these, 5-bromouracil and 2-aminopurine are the most commonly used ones. For example, 5-bromouracil is an analog of thymine that has bromine at the C5 position in place of the CH3 group found in thymine. This change does not affect the atoms that take part in hydrogen bonding in base pairing, but the presence of the bromine significantly alters the distribution of electrons in the base. The normal structure or the keto form of 5-bromouracil pairs with adenine as shown in the figure. 5-bromouracil can frequently change to either the enol form or an ionized form. The latter pairs in vivo with guanine. Thus, the nature of the pair formed in replication will depend on the form of 5-bromouracil at the moment of pairing. Here in this figure you can see 5-bromouracil causes transitions almost exclusively. This figure describes the mechanism of 5-bromouracil mutagenesis. 5-bromouracil causes mutations when it is incorporated in one form and then shifts to another form. In its normal keto state, 5-bromouracil pairs like thymine. Thus, 5-bromouracil is incorporated across from adenine and subsequently mispairs with guanine, resulting in AT to GC transitions. In its ionized form, 5-bromouracil pairs like cytosine to form 5-bromouracil C. Thus, 5-BU is misincorporated across from guanine and subsequently pairs with adenine, resulting in GC to AT transitions. Another analog widely used in research is 2-aminopurine, which is an analog of adenine that can pair with thymine, but can also mispair with cytosine when protonated, as shown in the figure. Therefore, when 2-aminopurine is incorporated into DNA by pairing with thymine, it can generate AT to GC transitions by mispairing with cytosine in subsequent replications. Or, if 2-aminopurine is incorporated by mispairing with cytosine, then GC to AT transitions will result when it pairs with thymine. Genetic studies have shown that 2-aminopurine like 5-bromouracil is very specific for transitions. This figure describes the alternative pairing possibilities for 2-aminopurine, an analog of adenine. Normally, 2-aminopurine pairs with thymine, but in its protonated state it can pair with cytosine. Alkylating agents. Alkylating agents are the chemicals which can transfer their highly reactive alkyl groups such as methyl or ethyl groups to the phosphate groups or bases of DNA. Ethyl ethane sulfonate or EES, ethyl methane sulfonate or EMS, methyl methane sulfonate or MMS, dimethyl sulfonate or DMS, etc. are alkylating agents. Alkylation of the phosphate residues and bases produces substitution mutation, frame shift mutations and chromosomal aberrations in the following ways. Alkylation of the phosphate residues interferes with DNA replication and inflicts breaks on the sugar phosphate backbone. This may produce phosphate triesters and induce gene mutation. The alkylation product of guanine or 7-ethyl guanine often pairs with thymine, that is replacing adenine, producing a transitional substitution mutation. During the repair of alkylated bases, errors sometimes occur. This produces transitional and transversional substitution mutations or even frame shift mutations. Some alkylating agents bring about the cross-linking of DNA strands or even DNA molecules. This in turn prevents DNA replication, induces chromosomal aberrations and produces substitution mutation and frame shift mutations. For example, ethyl methane sulfonate, a commonly used mutagen, alkylates guanine in DNA, forming O6-ethyl guanine. During subsequent DNA replication, O6-ethyl guanine directs incorporation of deoxythymidylate, not deoxycytidylate, resulting in the formation of mutant cells in which a GC base pair is replaced with an AT base pair. In this figure, you can see the EMS alkylates guanine at the oxygen on position 6 of the purine ring, forming O6-ethyl guanine, which base pairs with thymine. 2 rounds of DNA replication of a strand containing ETG yields a mutant DNA in which a GC base pair is replaced with an AT pair. Deaminating agents. Nitrous oxide reacts with bases containing amino groups. It can change the structure of such bases by deamination. It can bring about the oxidative deamination of adenine, guanine and cytosine, producing hypoxanthine and uracil respectively, result in transitional substitutions. On the other hand, xanthine pairs with cytosine, producing a machine pair. Intercalating agents producing distortions in DNA. The intercalating agents form another important class of DNA modifiers. This group of compounds includes proflavin, acridine orange and a class of chemicals termed ICR compounds. These agents are planar molecules which mimic base pairs and are able to slip themselves or intercalate between the stacked nitrogen bases at the core of the DNA double helix. In this intercalated position, the agent can cause single nucleotide pair insertions or deletions. This figure shows the structures of the common intercalating agents such as proflavin, acridine orange and ICR 191. An intercalating agent slips between the nitrogenous bases stacked at the center of the DNA molecule. Hydroxylating agents. Hydroxylamine NH2OH is very specific in its action. It reacts mainly with cytosine and guanine and brings about transitions and mispairing. This produces hydroxylaminocytosine, which pairs not with guanine but with adenine. Thus, hydroxylamine induces a transitional substitution. Other chemical mutagens. They include nitrosoguanidide, formaldehyde, mustard gas or dichlorodethyl sulfide, hydrazine, hydrogen peroxide, etc.